everyone. Today we will be discussing metabolism of carbohydrates. So this is the overview of metabolism of carbohydrates. The glucose which is taken up from the intestine into the circulation. So this glucose will enter into the cell with the help of glucose transporters, and that glucose gets converted into glucose 6-phosphate. And that glucose 6-phosphate will undergo a process called glycolysis, and at the end of glycolysis the product which is formed here is pyruvate. So what is the fate of that pyruvate? That pyruvate can get converted into amino acids. It can get converted into acetyl-CoA by oxidative decarboxylation reaction, or it can get converted into lactate in certain tissues which does not have oxygen. Okay, so what happens to the excess of glucose which is taken up by the cell? Suppose if the cell has got adequate amount of energy from this process of glycolysis, what happens to the excess amount of glucose? The excess amount of glucose will be converted into glycogen by a process called glycogen synthesis, and it will be stored in organs like liver and muscle as glycogen. And whenever required, for example, in the fasting state and in starvation period, this glycogen will be broken down into glucose, and that glucose will be utilized by the cells for energy. What happens to glucose 6-phosphate? The minor pathways. For example, there are certain pathways which will result in formation of certain compounds like ribose 5-phosphate and NADPH. So these pathways will occur continuously in certain tissues which require these ribose 5-phosphate and NADPH for the proper functioning of the cells. So this is the overview of the metabolism of carbohydrates. First, we will see glycolysis. Glycolysis is otherwise called Embden-Meyerhof pathway, or otherwise called Warburg pathway. Glycolysis. What is glycolysis? Lysis means breakdown. The glucose is broken down. So this is a major pathway of carbohydrate or glucose metabolism. Why this pathway is happening? To provide energy. And it also provides intermediates for the other metabolic pathway. Why glycolysis is important in certain tissues? Because brain, it rely only upon glucose for its energy. So glucose is the primary source of fuel in brain. In red blood cells, it does not have mitochondria, so it depends only upon glycolysis for ATP generation. So where does this glycolysis occur? It occurs in cytoplasm of all the cells. Glycolysis can be divided into two phases. The one is energy investment phase where ATP is utilized, and the other one is energy generation phase where ATP is generated. Along with ATP, NADH is also generated. And in the energy investment phase, we have two phases. For example, preparation of glucose for breakdown. So that is called preparatory phase, and splitting phase where glucose is splitted into 2,3 carbon compounds. First, we will start with the energy investment phase. In that, we will start with the preparatory phase. So what happens to the glucose taken up by the cell? Okay, so all the cells have glucose transporters. So via the glucose transporters, glucose is taken up by the cell. Once glucose enters the cell, the enzyme hexokinase or glucokinase will convert glucose to glucose 6-phosphate. Okay, when glucose is converted to glucose 6-phosphate, this glucose cannot get out of the cell because it is trapped inside the cell as glucose 6-phosphate. So the enzyme required for the conversion of glucose to glucose 6-phosphate is either hexokinase or glucokinase. Kinase, it indicates kinases will transfer phosphate group to the substrate. From where it is transferring phosphate group? It is transferring phosphate group from ATP. So in this reaction, one ATP is utilized. So one ATP is converted to ADP, and phosphate group from ATP is transferred from ATP to glucose. So glucose is converted to glucose 6-phosphate. So I told you two enzymes, right? Glucokinase and hexokinase. So what is the difference? If you see where it is present, glucokinase is present in liver, but hexokinase is present in all the other tissues. And what is the substrate for glucokinase? Glucokinase, only glucose can be the substrate. For hexokinase, any hexose. It can be glucose, it can be fructose, it can be mannose. What about the affinity of glucose to this enzyme? Glucokinase has very low affinity to glucose, and hexokinase has more affinity to glucose. And this particular enzyme, glucokinase, production is induced by insulin and increased high levels of glucose, otherwise called high levels of glucose, is going to induce the production of glucokinase. What is the function of this enzyme, glucokinase and hexokinase? So glucokinase will come into action when the blood has more amount of glucose, but hexokinase can utilize glucose even when the glucose level is low. So this is the difference between glucokinase and hexokinase. So glucose has been converted to glucose 6-phosphate. Next, what happens to that glucose 6-phosphate? Next, here comes the enzyme phosphohexose isomerase. So hexose, that means 6-carbon atom, glucose is a 6-carbon monosaccharide, isomerase. So glucose is converted to fructose. Okay, so glucose 6-phosphate is converted to fructose 6-phosphate. We all know they are functional group isomers, otherwise called structural isomer. So with the help of enzyme isomerase, glucose 6-phosphate is converted to fructose 6-phosphate. The reaction which is happening here is isomerization. Next, what happens? Next comes the enzyme called phosphofructokinase. So kinase, it requires ATP, and from ATP it is going to transfer phosphate group to the substrate. Here the substrate is fructose 6-phosphate. So already one phosphate group is present in the fructose. You are going to add the second phosphate group. So the product formed will be fructose 1,6-bisphosphate. So fructose has phosphate group attached to first and sixth position. So in this reaction, one ATP is converted to ADP. So we have synthesized fructose 1,6-bisphosphate. So this is a very important step. It is the irreversible step, and it is the rate limiting enzyme of glycolysis, and it is a highly regulated enzyme of glycolysis. Okay, and it is an allosteric enzyme. We have phosphofructokinase 1 and 2. So what happens to the formed fructose 1,6-bisphosphate? Splitting phase. So what splits fructose 1,6-bisphosphate? The enzyme here is aldolase. So aldolase is going to split fructose into 2,3-carbon compound. Okay, by acting in this site, so aldolase is going to cleave fructose 1,6-bisphosphate to 2,3-carbon compound. First one is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So glyceraldehyde, it is a 3-triose aldose. Dihydroxyacetone is a triose ketose, right? Yes, so we have splitted fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Next, again, the next step is isomerization. Glyceraldehyde is an aldehyde, aldose, dihydroxyacetone is a ketose. So ketose is getting converted to aldose here. So what happens? The dihydroxyacetone phosphate gets converted to glyceraldehyde 3-phosphate. Okay, so it is a 3-carbon compound, so triose. It has a phosphate group attached to it, and what is getting converted to what? A ketose is getting converted to aldose. So the enzyme is isomerase. So what is the enzyme here? Phosphotriose isomerase that converts dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Next comes the energy generation phase. So from here on, we will be synthesizing NADH and ATPs. So it is called energy generation phase. Glyceraldehyde 3-phosphate will be acted upon by the enzyme glyceraldehyde 3-phosphate dehydrogenase. So dehydrogenase, in this step, one NAD plus is converted to NADH plus hydrogen ion. So in that reaction of dehydrogenation, glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. Okay, so one phosphate group is getting added to it. So glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. If you see in the first position and the third position, 2 phosphate groups are attached. So 1 and 3, so bisphosphoglycerate is formed. Next step is the first substrate level phosphorylation which is happening in glycolysis. We have 2 phosphates attached here. What happens here is one phosphate is removed from the substrate and added to ADP, and that ADP is converted to ATP. Okay, so this is 1,3-bisphosphoglycerate. The enzyme here is bisphosphoglycerate kinase, because kinase, it is going to transfer phosphate group from substrate to ADP. So what is formed here? ATP. ADP is converted to ATP, and 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate because one phosphate group is removed. So 3-phosphoglycerate is formed. This is the first substrate level phosphorylation which is happening in the glycolysis. So what is substrate level phosphorylation? If the ATP is produced at the level of substrate, so that is energy needed for the production of ATP comes from a substrate rather than from the electron transport chain. So we will be seeing about electron transport chain in the upcoming classes. So what happens is, if ATP is synthesized inside the mitochondria with the help of electron transport chain, then that is called oxidative phosphorylation. Okay, so when ATP is produced within the mitochondria with the help of electron transport chain, that is called oxidative phosphorylation. Substrate level phosphorylation means where the energy which is needed for the synthesis of ATP is derived from the substrate itself. Okay, here the substrate is 1,3-bisphosphoglycerate. This itself gives energy for the synthesis of ATP from ADP. So this is called substrate level phosphorylation. Okay, next what happens is we are going to shift the phosphate group. We are going to shift the phosphate group from 3-phosphoglycerate to 2-phosphoglycerate. Basically, we are going to change the position of phosphate group. Okay, so from third position, we are going to shift the phosphate group to second position. So within the same molecule, we are going to change the position of groups. The enzyme which catalyzes such kind of reactions are called mutase. In which substrate it is going to change? Phosphoglycerate. So the enzyme here is phosphoglyceromutase. So this enzyme phosphoglyceromutase converts 3-phosphoglycerate to 2-phosphoglycerate. So what happens to the 2-phosphoglycerate? The next enzyme, enolase, which is going to remove water from 2-phosphoglycerate. It removes water molecule and it results in the formation of phosphoenol pyruvate. Okay, phosphoenol pyruvate. What is happening here is dehydration, removal of water molecule. How water is removed? With the help of enzyme enolase. And this enzyme enolase is specifically inhibited by an inhibitor called fluoride. Okay, so fluoride is an inhibitor of the enzyme enolase, the only inhibitor which you see which has a clinical utility. We used to collect blood sample in fluoride tube for estimation of glucose, because whenever the blood sample is not centrifuged, if it is kept as a whole blood sample, glycolysis will happen inside in the RBC and there will be falsely low level of glucose. To avoid that, we used to collect glucose in fluoride tubes. So what is the mechanism of action of fluoride here? Fluoride will inhibit the enzyme enolase, so glycolysis will not happen. Okay, next the last step is formation of pyruvate. We all know pyruvate is an end product of glycolysis, aerobic glycolysis. So the phosphoenol pyruvate, which is formed in the previous step, will be acted upon by the other enzyme, pyruvate kinase. So this enzyme is going to transfer phosphate group from ADP to ATP. So one ATP is produced. This is a substrate level phosphorylation. Second substrate level phosphorylation, which is happening in glycolysis. So phosphoenol pyruvate is converted to pyruvate. Okay. Yes. So far we have synthesized pyruvate. So 1-fructose 2,6-bisphosphate molecule is converted to 1 molecule of glyceraldehyde 3-phosphate and 1 molecule of dihydroxyacetone phosphate. With the help of enzyme phosphotryosisomerase, the dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. So totally we have 2 glyceraldehyde 3-phosphate. So what we get finally is 2 molecules of pyruvate at the end of glycolysis. So what is the fate of that pyruvate? The fate of that pyruvate depends upon the availability of oxygen. Suppose if there is oxygen, that is under aerobic conditions, this pyruvate is further oxidized to acetyl-CoA. Okay, acetyl-CoA. With the help of enzyme pyruvate dehydrogenase, this pyruvate is oxidized further to acetyl-CoA, and that acetyl-CoA will enter into TCA cycle. Okay, tricarboxylic acid cycle. This is the fate of pyruvate under aerobic conditions. Pyruvate further gets converted to acetyl-CoA. Acetyl-CoA enters tricarboxylic acid cycle. So far we have converted 1 molecule of glucose to 2 molecules of pyruvate. So let us see what is the energetics of this pathway. Okay, so in the first step, hexokinase step, we have utilized 1 ATP. And in the phosphofructokinase step, we have utilized 1 ATP. Yes, that is in energy investment phase, we have utilized 2 ATP. And how much ATP we have generated in the energy generation phase? First, in the first step, we have generated NADH. Now you have to remember, when this NADH enters into the mitochondria and if it goes through the electron transport chain, it is going to generate 2.5 ATP. We will see in the upcoming classes how 2.5 ATP is synthesized, but as of now you have to remember, one molecule of NADH, if it enters into electron transport chain, it gives 2.5 molecules of ATP. So we have synthesized 2 NADH. So 2.5 into 2, it is 5. So totally we got 5 ATP in the glyceraldehyde 3-phosphate dehydrogenase step. And we have seen 2 substrate level phosphorylation. 1,3-bisphosphoglycerate kinase. So we have synthesized one, and the other one is pyruvate kinase. In that we have synthesized one. So 2 molecules of glyceraldehyde 3-phosphate will undergo energy generation phase. So 2 ATP is produced in each step. Right? So totally we have generated 5 plus 2 plus 2, 9, and we have invested 2. So out of that 9 we have invested 2. So totally 9 minus 2, it is 7 ATP. So the energetic of the aerobic glycolysis is 7 ATP. We have generated 9 ATP and we have utilized 2 ATP. So 9 minus 2, it is 7. Okay. So we'll move on to the regulation of glycolysis. So what are the important regulatory enzymes in glycolysis? Hexokinase, otherwise glucokinase. This is one important regulatory step. The very important rate limiting and highly regulated step is phosphofructokinase. This is the rate limiting enzyme of glycolysis. Right? Yes. The final enzyme, pyruvate kinase. This is also one of the highly regulated enzyme of glycolysis. How these enzymes can be regulated? There are 3 different types of regulation. First one is nutritional regulation. It depends upon the status of the person, whether he had taken food or he had not taken food. That is, whether if he is in well-fed state or starvation state. The other one is hormonal regulation. Insulin, glucagon, epinephrine. Okay, so the other one is hormonal regulation. The third one is covalent modifications. The other one is covalent modifications. Okay, so nutritional regulation. So what happens when you're taking more amount of carbohydrates? That means you're in well-fed state. You have just had your food. So what happens? So there will be more glucose. So glycolysis will happen more. So increased glycolysis. When there is more glucose. What happens when you're starving? We don't have glucose. The cell should not utilize glucose. The cell should conserve glucose and it should send to the circulation to maintain the mean blood glucose level. So when you're starving, glycolysis will not happen. The other way around, the synthesis of glucose will happen. So when we are starving, glycolysis will not happen. So in well-fed state, glycolysis will happen, and in starvation state, glycolysis will not happen. Hormonal regulation. As we all know, immediately after having food, there will be release of insulin in our body. So what happens? Immediately after food, there will be increased insulin. So the insulin is going to promote glycolysis. So when insulin is going to promote or increase glycolysis. What about the counter-regulatory hormones? So the actions and functions of glucagon, glucocorticoids are exactly opposite to insulin. So what are the functions of these hormones, glucagon and glucocorticoids? They are going to decrease the glycolysis because they are the hormones which are going to maintain the blood glucose level in starvation state. So whenever insulin is present or more, it is going to increase the glycolysis, and when there is increased level of glucagon and glucocorticoids, they are going to decrease glycolysis. Next, moving on to the important enzyme. PFK is phosphofructokinase, the very important and rate limiting enzyme, and there are certain allosteric inhibitors and activators. That means these compounds are going to bind to this enzyme, and these compounds are going to modify the activity of the enzyme. Inhibition means when these compounds are present, they are going to bind to phosphofructokinase and inhibit the activity of phosphofructokinase. When the cell has more amount of energy, for example, that means when the cell has more ATP, don't want to synthesize further ATP. So what happens that that ATP will go and bind to phosphofructokinase and inhibit that. And citrate, we will see in the upcoming classes, increased citrate is a sign of increased utilization of glucose and more energy. So it is a high energy state. So when there is more amount of citrate also, that means there is more energy or more ATP in the cell. So enough. So this citrate will go and act upon phosphofructokinase and inhibit that. Okay, when this is activated, when the cell does not have energy at all, that is conveyed by increased level of AMP. Because we all know AMP will get converted to ADP, ADP gets converted to ATP. So when there is more AMP, that means the cell does not have energy, so it needs energy. So glycolysis should happen. So AMP will go and bind to phosphofructokinase and activate phosphofructokinase. The other allosteric activator, very important activator, is fructose 2,6 bisphosphate. Okay, in the pathway we have synthesized fructose 1,6 bisphosphate. The other molecule, fructose 2,6 bisphosphate, this is an allosteric activator of phosphofructokinase. When this is present more, this is going to go and bind to phosphofructokinase and going to activate phosphofructokinase. Okay, so this is how phosphofructokinase is activity is regulated allosterically, either by allosteric activator or by allosteric inhibitor. What is covalent modification? That is also called reversible. This is a type of reversible modification. For example, this enzyme phosphofructokinase will be active when it is dephosphorylated. This enzyme will be inactive if it is phosphorylated. So any enzymes which are going to dephosphorylate phosphofructokinase are going to activate glycolysis. The enzymes which are going to phosphorylate phosphofructokinase are going to inactivate phosphofructokinase and thereby inhibiting glycolysis. Okay, so this is all about regulation of glycolysis. We have nutritional regulation, hormonal regulation, allosteric regulation, and covalent modification. That's all for today's class. We will be looking into anaerobic glycolysis and Rappaport-Lauberin cycle in the next class. Thank you.